Hello and welcome to today's live event for the COP30 Virtual Ocean Pavilion. My name is Katie Mitchell. I'm a policy analyst with the Global Ocean Forum. We're one of the co-organizers for this year's COP30 Virtual Ocean Pavilion, along with Plymouth Marine Laboratory and Ocean Generation. For today's event, I will be on hand for any technical support you may need, so please feel free to raise your hand if you need any assistance during the call. Today's session will also feature live translation brought to you by Wordly. I'm going to share an invitation now to all participants. So if you would like to utilize translation during the call, you can accept that invitation and translate it into your preferred language. On behalf of the organizing team, thank you to our panelists, our organizers, and our attendees for joining today's event. And with that, I will pass it over to Dr. Jamie Wilson to get us started. Thank you.
Thank you. I'll just share my screen. The right one. There we go. Okay. Hi, everyone, and welcome to joining us today to this live event. So my name is Jamie Wilson. I'm a lecturer in the Ocean Sciences Research Group here at the University of Liverpool, and I'm the lead organizer of this event today. And today we're talking about from cells to climate and what is the role of biology in a changing climate. So we've been undertaking oceanographic research here at Liverpool for over 100 years now. And today we want to give you the scientific perspective on the key theme of the Virtual Ocean Pavilion, which is that the biodiversity, ocean, and climate nexus. So this is the idea, the idea that biodiversity is affected directly by changes in climate, and also indirectly as climate affects the ocean. But that biodiversity is not just a passive player in that system. It can change in response to that climate and then impact the ocean and then impact the climate. So we have this very dynamic system created by this nexus, but understanding how that system behaves, going from those individual cells to climate, is not an easy task, because as scientists, we have to work across multiple scales of time and space, as well as different scientific disciplines. So today, we've got 8 scientists from across the University of Liverpool and collaborators in the Tara Ocean Foundation and the Marine Biological Association. And they're giving you the understanding of how this nexus works, and then giving you individual snapshots of the key challenges and the key new advancements in this scientific area. So as scientists working in this area, when we talk about biodiversity in the ocean, we typically think about plankton. So these are these tiny little organisms floating around in the surface ocean. They're ubiquitous across the surface ocean, and a group of these, the phytoplankton, are microscopic, they're single-celled mostly, and they're plant-like organisms, so they're photosynthesizing. So they're taking up carbon dioxide from around their environment, they're using sunlight to break it apart to make biomass from the carbon and releasing oxygen as a byproduct. And that is what we term productivity. So although these organisms are really tiny and microscopic, they have a presence on the globe and an impact at a global scale. And that happens because just the sheer volume of the cells photosynthesizing across the global ocean area means that they account for about 50% of the global productivity. So as an example of the scale of that productivity, what I'm showing you here on the spitting globe is observations from satellites of that productivity from plankton, and we can see that from space because the light changes, the changes in light from the green pigment when it's being absorbed through that green pigment of these tiny cells can be picked up by satellites. So you can see the annual changes and time changes of that productivity and how vast it is across the surface ocean. So that productivity underpins 2 key ecosystems at a global scale that impact us and our society. So most of that productivity, about 80%, gets eaten by other stuff, so zooplankton, and that forms the basis of food webs and ecosystems, and that ultimately supports the fisheries that we rely on for food on a global scale. So the rest of that material, so all of the dead stuff, all of the organic molecules, all of the remains of that biological activity in the surface ocean, it contains that carbon that we extracted from the environment, and it will escape the ocean and make its way into the deep ocean. And that fuels a reservoir of carbon in our planet, in the ocean, that can help regulate the carbon cycle and the climate of our planet. So for me as a scientist, one of the most fascinating things, and the reason I do this job, is that these are like super tiny organisms living on, days or weeks, and yet they have no concept of this vast planet they're part of, and yet their existence has this impact on our society and the dynamics of our climate hundreds of years into the future. And to me, that's just really fascinating, the reason why I do this job. So plankton in the ocean are responding to the warming climate as we speak, and we expect to carry on into the future. So as that surface ocean warms, our models suggest that plankton species will be migrating away from the equator where it's warmest towards the poles at about a rate of 35 kilometers per decade. And the result of this is that those ecosystems are reshuffling around and they're becoming more unstable because of that. So we expect big changes in just the amount of plankton. So we expect to be losing lots of plankton in the surface ocean, particularly in the equator. And we're also expecting big changes in that biodiversity of those ecosystems that they support. And in particular, the North Atlantic and the Arctic Ocean is where we really expect those biggest impacts on the plankton and the ecosystem functions that they provide. So the big question really is, what will happen to those planetary ecosystem services of ecosystems and fisheries and carbon storage as the climate warms? So to answer that is not very easy. So it's no mean feat. So we have this grand scientific challenge. And the reason for this is that we have plankton living on the order of days or weeks, interacting on the planet on the order of decades, centuries, and centuries plus. These organisms are absolutely tiny. They're less than 1 millimeter big, but the scale they operate on is across millions of kilometers squared on the planet. We also have to integrate as scientists across different disciplines. So we need to be talking to biologists and ecologists working with these plankton, but we also need to be thinking about physics, the chemistry, and the Earth system to integrate into that scale. We also need different approaches. So we need that pure kind of observational based, that pure science of going out there and looking at where those plankton are and what they are, as well as we need the kind of types of approaches like using computer models. So this is a really big challenge as scientists because we have to navigate across this whole range of scales. So at Liverpool, that's one of our specialisms as a research group. We really pride ourselves in covering that diversity of scales and approaches needed to answer this grand challenge. So the aim of this event today is to give you snapshots into this grand challenge and give you across that range of those different scales. So it's going to happen in 3 different chunks. So the first chunk, we're looking at how we go from plankton cells and diversity to the marine productivity. So our first speaker, Martin, from Tara Ocean Foundation, will give you information about the latest results from how we actually go out and observe this in a really novel way using the Tara Ocean Foundation's boat. Then our PhD researcher, Ross Gracie, will talk about how we go out and actually observe plankton by using tiny little floating robots that can take pictures of these plankton. And then Alessandro will finish up with a recorded video, since he's away at the minute, looking at how we scale that information about genes and organisms into predictive models. So we'll have a short Q&A session after that first block, and then we'll move into the regional perspective. So I mentioned that the Arctic and North Atlantic are the key areas where we expect those big changes. So Nova will be talking about the North Atlantic, and Claire Mahaffey will be talking about work in the Arctic Ocean. And Claire is also away in Greenland at the minute, so she'll be giving a recorded talk. And then lastly, we're going to scale up into that global planetary scale and think about the carbon system. So we start off with Rick Williams talking about the ocean warming and ocean stripes. Chris Hughes will talk about ocean physics, and then I will finish to talk about what we think will happen to biological carbon storage in the future. So I'd just like to say thank you for joining us again today, and I will hand over to the speakers to start off. So I will hand over to Martin.
Thank you very much, Jamie. I will just try to share my screen also. Can everybody see the correct slide? I don't see you anymore. Yeah, I think it's good. Well, thank you very much, Jamie. Good morning, everyone. So my name is Martin Alessandrini. I'm speaking.
Martin, we
Yes.
are not seeing your slides.
Oh, no. Wait a second. I know how I'm going to do it. Tag. If I'm doing it this way, can you see it?
Yes, now we see your slides.
Great. So good morning again. So my name is Martin Alessandrini, and I'm speaking as an advocacy officer at the Tara Ocean Foundation, and I'll try to share a bit some insights of the science that's been done on board the Tara schooner for over 20 years now. So the ocean, as we're speaking for within this climate COP framework, I just want to remind maybe a big general figures that most of you know. This ocean is really the living engine of the Earth and produces roughly half of the oxygen that we have on the Earth and absorbs around a third of our CO2 emissions. There's always discussion around the figures, but it gives us actually a general understanding of its role in the climate system. And these planetary scale functions are actually made possible by this very diverse community of microorganisms that we find and that we know in the ocean as plankton, ranging from viruses, from the smallest one at the limit with living organisms, to bacteria and microalgae and small animals. And each group performs unique roles in carbon fixation, in nutrient recycling, in oxygen production. And yet, despite their importance, actually, the knowledge that we have about this plankton is really recent and remain largely invisible. So our mission at Tara is really to work with a lot of different international laboratories like University of Liverpool and to make visible this invisible majority of the ocean so we can better understand and protect the ocean. So we've been starting this for 20 years now, partnering with a lot of different international labs. More than 200 in this institution have been boarding the boat to study globally this planktonic ecosystems. Our schooner has sailed through all major ocean basins, generating over 1,000 papers. Most of them have been published by third parties, laboratories, because everything that's been produced on board is then publicly shared. But this approach is very unique, and it's to have a global, standardized, ecosystem-wide approach to map this marine microbiodiversity, and then to share all the data that's been produced with open data for everyone to be using it. And so it all started with our first major expedition called Tara Oceans, which was the first global plankton survey, I would say, and it was the first global effort to systematically sample plankton all across the ocean, from the surface waters to the deep chlorophyll maximum. And what was completely new was everything that we found in the samples was new data, actually. So we found more than 200 million new genes, understanding a bit the role that they have in the ocean, finding more than 200,000 new virus, bacteria, also microscopic species discovered. And the real insight was really that the ocean functioning depends on this vast, previously unknown biodiversity of microorganisms that all the scientific partnering with Tara Ocean helped discovering in these major expedition. And so lately we've been expanding these research projects in a lot of different ecosystems. The first one was also Tara Pacific, trying to study these microbiodiversity for coral reefs also, and trying to understand their genetic and microscopic diversity. Mission microbiomes very more recently in 2021, 2022, and Alessandro, who's going to be speaking after me, also took part in this expedition to try to focus more on the diversity on specific ecosystem like Antarctica. And it's something that we'll keep on doing with next expedition, the one with the boat. Tara is going to be Tara Coral in next year, trying to study these coral triangle reef ecosystems. But we also have now a second boat that's going to be focusing on the Arctic with Tara Polaris 1, the first Arctic drift of our second boat, which is going to be studying these microorganism diversity in the Arctic. And so here you have all of our major expedition, around 13 expeditions in 20 years, trying to map really this microbiodiversity, how it's structured, how it functions, and how it also is impacted by climate change. And so with all this genetics and imaging, because it's the two main techniques that are going to be used to study this plankton, all the Tara scientific partners have helped reveal who these organisms are, what they are doing, and how do they respond also to the stress. Because as plankton represents the majority of ocean biomass, they lead to all different outcomes: carbon fixation, nutrient regeneration, oxygen production, and also the export of the carbon to the deep ocean. And all these are themes that are going to be addressed by all the scientists making this brilliant panel today that are going to be speaking after me. And so all this community composition actually determines the fate of the carbon and of the ecosystem in the ocean. But climate models today still represent biology with a few different generic plankton groups, but not assessing its entire, I would say, biodiversity. So there's a real need for biodiversity to connect and to be integrated as a very strong climate variable. And lastly, because it's I think the point that that's gathering all of us today is just not about producing and understanding plankton, but also to make it useful for decision making. And that's the I think the input that Tara Ocean is trying to bring with all the scientists that are collaborating with us. It's to have this society impact also for science, trying to inform marine protection strategies, science-based policy making, and thus climate also frameworks like the COP. By identifying all these genetic markers linked to carbon sequestration, to stress tolerance, I think we can better monitor ecosystem health, anticipate risks, and also guide conservation measures. So protecting the ocean means protecting the biodiversity that we have in the ocean, and mostly also this micro biodiversity and all the functions that help regulate the climate. So I leave it to this brilliant panel of scientists that's going to be following me to dive deeper into all these plankton functions and how these microorganisms and their very small-scale genetic and functions connect to global challenges that we're tackling at climate COPs. Thank you very much.
Well, thank you, Martin. So next we're going to pass on to one of our PhD researchers in the group, which is Ross Gracie. He's just sharing my office with me, so I'm just going to nip out so we don't mess up with any microphone and feedback. So over to you, Ross.
Thank you, Jimmy. Thank you, Martin. I'll just share my screen here. So, hello everybody. My name is Ross Gracie, and I'm a PhD student here at the University of Liverpool. Today, I'm going to be talking to you about imaging insights into phytoplankton communities. So, why is phytoplankton biodiversity so important? Well, phytoplankton are tiny ocean plants that capture energy from the sun through photosynthesis. They turn the sunlight into food, transferring the energy up the food web, supporting fish, whales, and all ocean life, and ultimately us. But photosynthesis just doesn't mean food. It controls the air that we breathe. And this process provides oxygen, supporting life on Earth and within the oceans and seas. Plankton also play a huge part in mitigating the impacts of climate change. Through photosynthesis, they take in carbon from the atmosphere and store it inside their cells. Whenever they die, they sink down the water column, storing this carbon in the deep ocean for a very long time through a process called the biological carbon pump. So, how do we actually study and research plankton in our oceans? Well, in the past, we've monitored plankton typically using 2 methods. Firstly, research vessels would drop nets into the ocean, and plankton would be collected and then counted by a scientist manually with a microscope. This means that we could get really high taxonomic resolution of what was actually in the samples, but we were limited by time and space from where the research vessel established stations. Secondly, we also used satellite imagery to see where the blooms were present. So this showed us where the blooms were, how long they stayed, but we couldn't exactly see what was in the blooms and what made them up. But what if we could collect high resolution plankton data without these spatio-temporal limitations? Well, somebody asked this question and decided that it would be a good idea to make an instrument to tackle these limitations. So I introduced to you the imaging flow cytobot, or the IFCB. So the IFCB is an automated in-situ plankton imaging classification instrument located in the ship's underwear. So as the research vessel moves through the water, plankton samples are being collected continuously. This combines flow cytometry and video microscopy. So as the sample enters the IFCB, the water is essentially stretched out with the cells entering single file. And as the particles pass through, laser light is scattered, and if chlorophyll is present, some of the laser beam is reabsorbed and readmitted, and this captures the images of the particles. So this images anything containing chlorophyll within a size fraction of 10 to 150 microns and measures up to 200 parameters per each organism. So we have all of our images collected not thanks to the IFCB, but how do we process these? Well, at the University of Liverpool, we use machine learning software called ecotaxy, and this improves the efficiency and automates the process of verifying these images. So the data is fed into the software where user-built algorithms predict the image to high taxonomic resolutions. After the predictions, the user will then validate the data, and this can then be exported for analysis. This then allows us to have species-specific identification in real time due to the help of machine learning, and it can accurately predict these images. So, as we export the data, we may expect to produce a figure that looks something like this. You can see how this tidally combines phytoplankton groups of all shapes and sizes, such as diatoms, dinoflagellates, some haptophytes, and even unclassified plankton can also be categorized into the bars. So this allows easy comparison compared to the endless pages of images. But how might we use this in the real world? Some of you may ask, well, these figures can be applied to the real world in conjunction with environmental data, such as what we call bottom-up controls, such like water, nutrient levels, or seafloor depth, or even what we call top-down controls, such as what predators are present in certain regions. And we can see how these effects cause certain groups to bloom or decline. We can also use the images to get insights into the phenological patterns showing where species might thrive or decline in populations. And the images also allow monitoring of species and size-specific structures of communities, which can be transferred then to Earth system models, allowing estimates of carbon flux linking phytoplankton to global ecosystems. But how can we all use this in the future? What can we use this data for? Well, if we take the images from the IFCB of our phytoplankton organisms, and we pair this with the biogeochemical Argo float data that measures things like pH, temperature, or fluorescence, as well as other autonomous sampling networks such as particle capture technology, it will actually help to give us a better idea of how ocean processes interact, and thus giving higher resolution to Earth system models. And this can potentially improve climate predictions. But why is all this important? So bringing it back in summary, we can use this state-of-the-art technology to detect and quantify the presence of certain plankton groups to various environmental conditions within our oceans. This means that we can use the community composition data to estimate and predict plankton's impact on the carbon export within the ocean, down to a species or size structured efficiency. This information can then be fed to Earth system models, improving climate predictions, and this can help us make better decisions about how to mitigate the effect of climate change. If anybody has any more questions on this, please feel free to contact me with the email address on screen. And whenever, I'd like to thank you all for listening. And whenever we get to the question break, I'm happy to take on any questions from the slides. Thank you.
Cool. Thank you, Ross. And now, so the final speaker, we have Alessandro Tagliabue. So he's going to be talking about how we translate from that diversity into climate models. So Alessandro is currently heading up a meeting about this exact stuff. So he's prepared a talk for us, which I'll just share now. Okay. I think we can see that, Katie.
Yes, we can see it.
Great. Okay. So this is recording from Alessandro talking about.
Hello everybody, my name is Alessandro Tagliabue and I'm a Professor of Ocean Sciences in the School of Environmental Sciences at the University of Liverpool. Today I'm going to talk to you about marine productivity and the grand challenge of being able to predict how it will change in response to climate change. In the snapshot you can see on the screen a representation of ocean fertility. The green areas represent those areas of the ocean that are particularly fertile, and the blue areas represent the oceanographic deserts where rates of productivity are very low. You can see lots of strong gradients in the ocean between different regions, across oceanographic features, and between hemispheres. Ultimately, we need to understand the mechanisms that shape this variability, because with that understanding, we'll be better equipped to predict the response of marine productivity to climate change in the future. Ocean productivity is important because it supports fisheries and the food supply. Primary production in the ocean is performed by microscopic marine phytoplankton, who use the energy from the sun to fix carbon and generate fuel for marine ecosystems. This is important because the fish that are supported within ecosystems make up a key source of protein for society. Future predictions, illustrated in the map you can see on the right, show reductions in the fisheries' catch potential, which would threaten ecosystems, but also food supply, because it's in the exclusive economic zones that show the largest reductions in catch potential that are bordered with nations that rely on the ocean to provide the protein for their food supply. However, at present we have a problem because our projections of how ocean productivity is going to change are too divergent to be useful for policymakers. On the left you can see model forecasts for changes in sea surface temperature, and you can see very clearly how these predictions separate between the low emission scenarios in the blue, through the green, orange and to the red of the high emission scenarios. This is really useful for policymakers because it allows them to link choices around emissions to outcomes in terms of sea surface temperature. On the right, you can see the same kinds of information, but this time for predictions of how net primary production of the ocean will change. And here you see a very different message. All of the different coloured lines overlap with each other, and the uncertainty around each multi-model mean is very, very large. Put simply, we cannot even predict whether productivity will go up or down. This is important because predictions of how changing fish stocks will react to climate change rely on having sufficiently confident projections of how climate change will affect productivity. At present, we simply don't have this information to provide them. Better constraints on the functioning and sensitivity of phytoplankton physiological processes is critical. Marine phytoplankton and the link between changes in the environmental conditions, such as the availability of light and key nutrients, and their ultimate productivity is driven by their internal physiology. This internal physiology is an ensemble of metabolic reactions that govern the vitality of the cells. Currently, Earth system models make very simple assumptions about how this physiology operates, and these assumptions ignore the advances that we've been making as a community in genomics and systems biology. At Liverpool, we're trying to pioneer the use of so-called genome-scaled models that are then coupled to Earth system models to improve confidence in projections. These genome-scale models work by directly exploiting genomic data. This genomic information, based on phytoplankton gene sequencing, reveals the ensemble of intracellular reactions. We then use the environment taken from a global model, which might consider the light and the availability of different nutrients, which then drives the internal physiology through the metabolic pathways and machinery. What's important is that these genome scale models are free from assumptions about parameterization, since they're reliant on data, and produce outputs such as growth and productivity, as well as the production of other metabolites of interest. What's particularly useful is that these genome scale models allow us to move beyond the state of the art and represent the production of different internal carbon pools. Here I've just shown an example of lipids and glycogen. On the left, we can see how the abundance of lipids as a function of biomass is strongly controlled by the temperature range the organisms are growing in. On the right, we can see how the glycogen, and particularly the storage of glycogen, and whether cells are depleting their existing store of glycogen or whether they're building up a store of glycogen, is linked directly to the light exposure the organism is experiencing. This is important because this represents the nutritional quality of phytoplankton, which is then fed up through the trophic levels of the ecosystem and of direct benefit to efforts to model the evolution and dynamics of marine fisheries. Looking to the future, we want to reduce uncertainties further, and to do so we are working on developing the concept of ocean systems models. These ocean systems models would exploit a suite of genome scale models tailored for each critical key phytoplankton group. This would allow us to represent the biodiversity at an ecological level, but also at a physiological level. This would be really important because it would reduce the need for these ad hoc parameterizations that models currently rely on, and therefore improve confidence in predictions. These ocean systems model would generate mechanistic coupling to upper trophic levels, useful for fisheries, as well as representing the feedbacks on carbon and nutrient cycling, useful for understanding climate regulation by the ocean. Ultimately, these ocean systems models would help us develop much more confident forecasts useful for policymakers and others interested in managing and mitigating climate change impacts on the ocean. With that, I will thank you for listening and for your attention to this webinar. Thanks.
Cool. Thank you to Sandro for preparing that for us earlier. So that concludes the first part of our trip along the sails to climate. So going from plankton diversity and how we scale that into predictions of productivity. So we have a couple of minutes for the panel to answer any questions you might have, and just to have a quick comfort break. So the panel will be here to answer any questions, and we'll get going with the next talks in a couple of minutes.
And to our audience members, if you would like to submit a question, please use the question and answer box that's available at the bottom of your screen.
So we have a question about what's the panel's opinion on banning bottom trawling. I wonder if, Nova, would you have any perspective on this?
Banning bottom trawling. I think that that's something that's already happening in some marine protected areas, not all marine protected areas. And again, it's a question of balancing the health of ecosystems and the species that are involved with human need for feeding themselves and sustainability. So it's a difficult question. I think we can still go further to maybe look at areas to definitely ban bottom trawling and focus it in areas where species aren't getting fished out so much and other species that aren't the target of the trawls are getting less damaged. But it's definitely something that I think we need to do further work on.
Okay, so I think for interest of keeping on time, I think we'll get going with the next segment on sort of the local perspectives. So we're going to have a little shift now, a little interlude, talking about particular areas of the ocean and what's happening to the diversity there and how that links to the changes in productivity and carbon. So I'll hand over to Nova to start us off.
Hello, everybody, and welcome to my section of the talks today. And I'm going to talk to you about how we track climate-driven changes in marine biodiversity and ecosystem functioning in my section of the world up in the Northeast Atlantic. Now, some of you may or may not know, I hope a lot of people have felt it, that the regional seas around the UK and Northern Europe have become warmer and warmer in successive years. And you can see that this year has been an incredibly warm year, according to the long-term records. We're also, as well as getting this pervasive warming of the marine climate, getting marine heat waves occurring a lot more frequently. And these are very, very short, acute hot weather events as well, which are very different to long-term warming of the climate, but can also double down and exacerbate climate change impacts on our marine species. Now, I run the Markland project, which looks at marine biodiversity and climate change. And this is a UK-wide project which was looking at bringing together historical data that has been collected since the 1950s by some very insightful marine biologists and current data for intertidal rocky shore species. These are excellent sentinel species of climate change because they're all stuck to a rock or can't move very far. Therefore, they have to adapt or they perish. So the first thing we did is repeat the classic broad scale surveys that were done way back almost a century ago, and then established a long-term network of 100 sites around the UK and France to monitor change. We then forecast future change based on using these data and the United Kingdom climate projections and the NOAA data, and then we push out this information to the people that really need to know. So we help direct policymakers on the marine environment and help them address the implications for society. This is Markland in a nutshell. You can see on the left all of our annual sites that we survey and the species that are showing the most change. Now, Markland is now the most spatio-temporally extensive intertidal time series globally, thanks to these really, really insightful people that started to notice changes way back in the early 1900s. And we're seeing species shifting along a biogeographic transition zone that bisects the UK from the warmer Lusitanian waters to the south, where these species are extending their leading range limits. And unfortunately, quite a lot of our boreal cooler water native species are starting to retract their trailing range edges. The two middle graphs just give you a little bit of indication of how we're quite confident. It's a large, long-term change. The top site there is my favourite species of topshell Phorcus lineatus at the northern range limit. And you can see that every single year throughout my venture through Markland, it's been increasing. So this is a long-term change. And on the bottom shows the same species across from this site all the way down into mid-France. So to hopefully show you again, since the 1980s, where the paler bars are, you can see that across a large geographical scale and latitudinal spread, this species is massively increasing in abundance at all of these sites. And because we've got fantastic sea surface temperature data that's now at a spatial resolution that's almost site specific, we can start to link this to climate change. We also carry out experiments so we can look at the biological mechanisms that are driving all of these changes. We've seen that every single species is changing in a different way and at a different rate. So we wanted to know exactly what's happening. Because of this fantastic time series, we have data going back to the 1950s, as you can see in the graph at the bottom. The bars show where there's been cooler than average years, sea surface temperature wise, and the red bars are where it's been warmer. No surprise, it's been warmer for a long, long time now. But you can also see then that the boreal barnacles, the blue line, have done better in previous years when it was cooler, but unfortunately now are massively declining in abundance, whereas the warm water barnacles are starting to take over. So we're seeing really, really big shifts in these species, which are echoed across the intertidal. And because we've got this wonderful time series, we can also look at extreme events as well. We've seen that there's been an increase in storm events since the 1970s. And certainly in the UK, in the winter of 2013-14, we had a storm event that was unmatched in intensity for over half a century. We had the biggest wave heights, the most rainfall, but we didn't have any extreme temperature events. And because we've got this beautiful time series, we're able to go back out again, as you can see in the two pictures in the bottom, and go and see what happened before the storm event happened and then afterwards. What we found here was every single site we surveyed had different impacts. Some were biological, either loss of species or gain of species. Some of it was habitat destruction or uncovering. And we found a really, really big difference, and it was site specific, which again shows the need for lots and lots of different records at different sites. And we also then use this to look at niche tracking using something called the Community Temperature Index. And this basically identifies the prevalence of whether warmer water species are doing better and how cold water species are declining. And it tracks the poleward distribution shifts and expected abundance. And what we need for this are long-term monitoring programs such as Marklim, so we can look at the changes in abundance of all the species in every community that we study and how it's affected by warming. We've done this for Marklim, and you can see that we found a real difference in the invertebrate animal species compared to the seaweeds. So the different taxa are responding differently. The middle two panels show that, again, even in a somewhat small area as the UK, the southwest of Britain was showing completely different responses to up in the north. And then the third panel with the full composite shows this was reflected in the community temperature index, thermal index, which obviously got cooler as we moved to communities further north. And this is a really nice way of pulling all of this information together and looking at how communities are changing. And we then expanded this idea to across Europe, and this just wasn't for rocky intertidal species. This was benthic time series, this was pelagic plankton and fish time series. We brought all of these different wonderful time series together to look at what the effects were across Europe and across different ecosystems. We had over 1,800 species from 65 time series that covered over 4 decades. And all of these time series showed the same thing, that when the sea surface temperature was increasing at between 0.32 degrees centigrade on the surface down to 100 meters at 0.15 degrees C, there was a significant trend. So warming in the waters, therefore, showed an increase in the community thermal index of all of the species as well. And this really brought home there are broad scale impacts of climate change that we're seeing across the North Atlantic seas, not just around the UK. The Marine Biological Association and the University of Liverpool have a really strong science policy remit. So all of these data and results are pushed out at the national level to an organisation called the Marine Climate Change Impact Partnership, where this takes science and it pushes out to policymakers in an easy to understand message. And then also for the European Marine Strategy Framework Directive and the UK Marine Strategy now, we're able to develop good environmental status indicators to show how all of these species are changing and how they're linked to climate. And the next steps, well, we need to keep the funding going for these long-term time series because they're vital to be able to show change across this large time series. But also we need to start to look at maybe initiating funding in areas that are lacking in data. So South America, Africa and Asia have an awful lot less long-term observations, and it would be really fantastic if we were able to initiate them with some long-term funding. We also need to look at how we're not seeing linear responses of all of these species, and we need to then start to look at bringing in experimental data to supplement these sustained observations so we can do some really good new mechanistic distribution models. However, these all need data. So I really want to put this out to the global audience. We need a global call to continue to fund existing programs and also to try and start up some new monitoring programs in areas of the world that don't have data at the moment, and work with people in AI as well to start to look at auto classification of biota, because this may be one way where if we can't survey everywhere, we can at least get images and photographs, and then we can start to look at how we can develop AI as a tool to help us address these knowledge gaps. If anybody's more interested in this and loves rock pooling as much as I do, then please get in touch. And I'm going to now hand back to Jamie.
Cool, thank you, Nova. So we've got a couple of questions for you that have come through, but we'll save them for the session straight after the next talk. So next up, let me just get it ready. So we have a talk from Claire Mahaffey, who is a professor here in our Ocean Researchers group. Just trying to find it. See him. Okay, and she'll be talking about nutrients in the Arctic Ocean. So Claire is currently away in Greenland conference, so she's kindly recorded this in advance for us. So I'll just play that for you.
Hello, my name is Claire Mahaffey. I'm a professor of ocean sciences at the University of Liverpool. Today I'm going to talk about the Arctic Ocean nutrient problem, asking the question, how will future primary productivity be sustained in a changing Arctic Ocean? So the Arctic Ocean is the smallest, coldest and freshest of all our oceans. The Arctic Ocean receives, circulates and redistributes water between the Atlantic Ocean, shown in red arrows, and the Pacific Ocean, shown in green arrows. This connectivity with the Atlantic and Pacific means the Arctic Ocean plays a vital role as a global climate regulator, influencing weather patterns and ocean circulation far beyond the polar region, including in the UK. The Arctic Ocean is also a hotspot of biomass and productivity and contains some of the most productive waters in our global oceans. It is home to ecologically unique, cold-adapted species that thrive in some of the harshest environments on Earth. What happens in the Arctic does not stay in the Arctic. Its health is critical, not only for the Arctic communities and ecosystems, but for the stability of our global climate, our fisheries, and also the societies that depend on them. A key characteristic of the Arctic is sea ice. Sea ice expands and contracts with seasons, shaping ecosystems and regulating climate by reflecting sunlight back into space. But the Arctic is warming 4 times faster than the global average. Under worst-case scenario projections, this means by 2100, the Arctic will have warmed by 10 degrees Celsius compared to a global average of around 4 degrees Celsius. A consequence of this warming is ice melt, and indeed the areal extent of sea ice has declined by about 12% per decade since the 1970s. Sea ice decline has allowed more sunlight to penetrate into the ocean with consequences. The Arctic Ocean is getting warmer due to the extra heat, but also microscopic plants, known as phytoplankton, are receiving more sunlight. As my colleagues have already introduced, phytoplankton are essential for supporting marine ecosystems and cycling carbon. To grow, phytoplankton need 2 things, light and nutrients. The extra light penetrating into the surface of the Arctic Ocean has caused primary productivity in the Arctic to increase by around 57% over the past few decades, as shown in the top left panel, with hotspots in the Barents Sea and Chukchi Seas, as shown in the red circles. As the Arctic continues to warm and sea ice retreats, numerical models suggest that productivity will keep rising, but with a high degree of uncertainty, as shown in the bottom left panel. This high uncertainty is driven by our lack of understanding on how a key nutrient resource is supplied to the Arctic Ocean, specifically the nutrient nitrate. We have traditional thoughts of how nitrogen is supplied to the Arctic Ocean. We have inflow and outflow between the Atlantic and Pacific Ocean. The inflow supplies about 18.8 teragrams of nitrogen per year. Outflow from the Arctic to the Atlantic and Pacific supplies about 18.4, which means we already have a deficit, that is nitrogen loss, within the Arctic. There's a small input from land and the atmosphere, and also rivers and coastal erosions, but that is a fraction of the oceanic source and is around up to 3 teragrams of nitrogen per year. However, there is a strong demand of nitrate in the Arctic through removal in sediments by a process known as denitrification, and also via increased demand by phytoplankton as productivity increases. Overall, the removal or sink of nitrate is greater than the nitrate supply into the Arctic, and this leaves a deficit in the nitrate supply, allowing nitrate to become the nutrient that limits primary productivity in the Arctic Ocean. This problem is exacerbated by the fact that we have strong evidence that nitrate concentrations have decreased in the Arctic. Research conducted at the University of Liverpool using numerical models has shown that nitrate concentrations in the Fram Strait have declined by 2 micromolar between 1970s and 2020. This is shown in the panel on the left-hand side. This model result is supported by a synthesis of observations led by scientists at the Scottish Association of Marine Sciences and Liverpool that illustrates also a measurable decline in nutrients, as shown in the panel on the right. This decline has been driven by changes in ocean circulation and by increasing biological demand as phytoplankton take advantage of warmer, ice-free waters. These shifts highlight an emerging paradox in the Arctic. While light has increased, allowing phytoplankton to thrive, the essential nutrients that fuel Arctic productivity may be running short, with major implications on the region's ecosystem and the global carbon cycle. So this begs the question, how will nutrient supply keep up with increasing productivity in the future Arctic Ocean? A key part of the solution may be nitrogen fixation, a process we once believed to happen mainly in the warm, low-latitude ocean. Biological nitrogen fixation is the global source of nitrate to the ocean. There has been decades of research on nitrogen fixation in the low-latitude ocean, with each observation illustrated in the global map on the in red. Nitrogen fixation adds new nitrogen to the surface ocean and relieves nitrate limitation for phytoplankton. We thought the organisms driving this process, known as diazotrophs, were restricted to the warm, low-latitude subtropical ocean until recently. We now have strong evidence from both the use of genetics and biological assays that diazotrophs are present and active in the Arctic Ocean. Measurements are sparse, but if we upscale them, we can estimate that nitrogen fixation could offset the deficit by denitrification by around 24% and support around 10% of productivity, meaning our current understanding is that nitrogen fixation could be a meaningful source of nitrate to the Arctic Ocean. As the Arctic continues to warm, nitrogen fixation may increase, thus providing an increasingly important nitrate supply to support productivity. Nitrogen fixers or diazotrophs found in the Arctic are diverse, and some may be associated with phytoplankton hosts, such as coccolithophores. Thus, our discovery of nitrogen fixations in the Arctic is not only important as a new source of nitrate, but also contributes to the biodiversity of phytoplankton communities in the Arctic Ocean. At Liverpool, we are leading expeditions to the Barents Sea in the eastern Arctic Ocean and the fjords in western Greenland to study the diversity, activity, and drivers of Arctic nitrogen fixation. Our goal is to collect new high-resolution observations, develop mechanistic understanding to improve numerical models on how nitrogen is supplied to the Arctic Ocean, and also how this nitrogen can sustain Arctic productivity. Finally, climate change is driving rapid and unprecedented transformations to the Arctic Ocean ecosystem, including to its productivity. There is an urgent need to understand how productivity will be sustained, with consequences for carbon cycling and marine biodiversity. With the 5th International Polar Year on the horizon in 2032, we have an opportunity to accelerate international, collaborative, and meaningful research in polar regions. Thank you very much for listening.
Cool. Thank you to Claire for recording that in advance for us. So we've got another short break, comfort break, and break for some questions. And there's a question for Nova that came through that I'm not going to attempt to pronounce the name of that species. So I'll let you take over.
Thank you very much. So the question comes from Michael Paulston, and it asks, do I think that the boreal cold water barnacle that has its retracting range limits in southwest Britain, Semibalanus balanoides, do I think it could entirely disappear from southwest coast of England? Yes, I do. And that's the beauty of having our long-term time series and going out every year, is I've seen the numbers declining, even just in my short 24-year tenure leading these surveys. And some sites now where it used to be the most abundant species in the low shore, I can hardly find it at all. Now, this is in just my short time looking at climate change impacts is really, really evident. So I do think it will entirely disappear, and I don't think it'll be beyond my retirement age, unfortunately, either.
Deborah, I was wondering how much do you see about species moving around parts of the coast rather than sort of disappearing on the spot?
So there's some are doing better. Obviously, those that prefer warmer conditions are increasing their latitudinal ranges north because the seas are becoming more suitable for it's mainly the juvenile stages to survive. But as I say, every single species is responding differently, which is why we need to keep getting out and consistently monitoring them, because we can't just do some kind of forecast model for the entire community as a whole, unfortunately. And also on top of that, we're seeing heatwave events. Until this year, they don't seem to have had any major impact on the rocky intertidal fauna up in our part of the world. However, we saw really, really extreme evidence this year for the brown algae for the fucoids. And I'm very interested to see when we go back next year, whether for the first time we're going to see major heatwave impacts on some of our macroalgal species that are community and ecosystem engineers and very functionally important.
For the people who are watching, the weather outside in Liverpool currently is completely drizzly and rainy and cold. So it's very funny to hear Nova talking about the northwest of the UK warming up.
Well, that's true, because if you remember, if any of you watching are as old as me, you remember that in November in the Northern Hemisphere, certainly in the UK, it'll be snowing, and quite often in the northwest of England would be stuck in with snowdrifts. So that's a beautiful example, Jamie, of climate change, because instead of snow, we've just got rain now.
Got rain. Cool, thank you, Nova. So I think it's reached 1 o'clock now, so I'm going to start on to the third section of the talk, so the final section. So in this section, we're going to think about moving up towards a global perspective, integrating across warming, ocean physics, and then into the carbon cycle. So I'm going to hand over to Professor Rick Williams to start talking about the ocean warming.
Thank you, Jamie. I'd like to turn to the question of what's happening on the planetary scale for the system. Space. And this is work with Ed Hawkins and a team of collaborators, where we've been looking at how the planet is warming at different levels through the atmosphere and the ocean. So we're all aware of the upward rise in global surface air temperature, reaching pretty close to 1.5 degrees. And we're used to seeing that as scientists in terms of an axis measuring the size of the change, and then the horizontal axis is giving us the time. What Ed did was communicate this in a superb way to the public, where the coloured stripes represent dark red represents the warmest year, dark blue represents the coldest year. So visually, you get this picture of warming stripes conveying what's happening to the planet. This is a particular picture for the global surface temperature, but he's done this for different cities throughout the world. But that then begs a question: how does that surface representation of climate change connect to the rest of the atmosphere and the ocean? And that's what I'm going to address here in a study that we recently wrote up in BAMS. So firstly, you can look at the troposphere, the lower 10 to 15 kilometers of the atmosphere. And what we see now are horizontal expressions of the warming stripes, where we've evaluated the warming stripes for each of those levels. And the key point is that over this time frame, now from 1980 to 2024, we're seeing at each level of the troposphere that the more recent years are the warmest of the past 40 years. So this is consistent with what we're seeing for surface air temperature. Then if we look up in the stratosphere, we actually see a cooling. This cooling of the stratosphere was actually predicted by Manabe in some of the first climate models, including greenhouse gases. What we see is the gain in heat in the troposphere through the presence of carbon dioxide is changing the heating profile, so we see a loss of heat in the stratosphere. That is all consistent with the planet gaining heat overall. There's also a lesser contribution from decreased absorption of solar radiation from depletion of ozone. But this structure is a classical fingerprint of climate change from greenhouse gases. Then how about the ocean? The upper 2 meters of water holds as much heat as the whole of the overlying atmosphere. And if you extend the warming stripes down into the ocean, this is extending. Different levels here are for levels of 100 meters, 200, 500 meters, 1,000, 1,500 meters. We actually see consistently that the more recent years are the warmest on record. So what we're seeing at the surface is carrying over to the record at depth in the ocean. Now, this is important because the bulk of the heat is stored in the ocean. And if we work out the global ocean heat storage over the upper 2,000 meters, we again see that the most recent years are the ones that are warmest. Now, that picture in the same way that local surface air temperature varies by location, that does vary by location. That climate warming signal is particularly striking in the Atlantic Basin and in the Southern Ocean Basin. Both of these basins are very well ventilated, have very strong communication with the atmosphere via the mixed layer. The Indo-Pacific Basin, we're seeing much more variability there. So what we're seeing in entirety is this picture for how the planet is warming. At all levels in the lower atmosphere and the ocean, the warmest years are typically in the most recent decades. But there is a cooling in the stratosphere, and that's due to the effect of CO2 in altering the absorption of radiation with height. So finally, what does the future hold? And that's really what this COP process is all about. It really depends on the scenarios that we choose to follow. In terms of the warming stripes, if we continue on our present path, we will be experiencing more extreme temperatures, and that will manifest itself in terms of regional changes in temperature in our environment that our previous speakers have been drawing upon. Or we can adopt more rapid action and try and move throughout the world towards the net zero aspiration. And that way, it will have this lower picture in terms of warming stripes. Okay, thank you.
Cool, thank you, Rick. So we'll pass on next to Chris Hughes, who's a professor here in the Ocean Sciences Group in Liverpool, and Chris will be talking about sea level and ocean physics.
Thank you, Jamie. So Rick's been talking about global, but he did speak a little bit about regional patterns as well. That heat that's going into the ocean is causing sea level rise, and it does so globally. But sea level rise is not all global. It can be regional as well. So what happens locally can actually have global effects and vice versa. We currently expect sea level to rise globally by at least tens of centimeters, possibly meters, with major impacts of coastal flooding. But these sea level differences from place to place can also change, and also by tens of centimeters. When we look at what the models do, they predict very different dynamical parts of the sea level change, and we don't know why. And what I want to talk about here is some work we've been doing recently trying to explain that in terms of ocean dynamics. So here's a picture showing what mean sea level looks like. You can see much higher sea level up in the Pacific, much lower sea level around Antarctica, 2 to 3 meters range, depending on where you're looking. But you can also see from this that the Atlantic and the Pacific are at different heights. Now we understand from theoretical arguments how these gyres come about that produce these higher sea levels on the west. But we develop that solution from the east, from a given value. And if we look at just the eastern sides of the ocean basins, you can see that starting value is quite different in the Atlantic and Pacific by sort of 20 to 40 centimeters, depending on how you define it. Now, the thing that causes that is mostly density in the upper layer of the ocean. So these two graphs here are for a whole series of CMIP6 climate model runs. They show the sea level difference between the Atlantic and Pacific on these eastern boundaries. at the start of a run, and then the difference between start and end. And you see the observed value about 29 centimeters. These models give quite a range of different predictions for the current state. And they also give a very large range of predictions for how things will change in the future. So the question is, can we explain these differences between the models? And one thing we can say is the other axis on these graphs is the part of that sea level difference which is due to changing density. So you see that's basically explained by density changes in the upper ocean. So it looks as if this is an upper ocean process, but we'll see it's not. Now it's hard to work in terms of sea level when you're trying to understand the dynamics. It's actually much easier if we think in terms of pressures on the ocean boundaries. So this is the theoretical framework that we've been using. Pretty crude picture for a pretty crude system. It's very simple. All we're saying is that for there to be a force balance, the winds have to be balanced by pressure forces on the boundaries of the ocean, unless there are currents in certain places that allow for momentum to be transported, or angular momentum to be transported. And the summary of this picture is, if you want to know the difference between the pressures along the boundaries of the Pacific and the boundaries of the Atlantic, all you need to know is winds along these colored lines and the deep flow underneath the red lines. So that's the flow into the Pacific and Indian Oceans below 1350 meters, which is the depth below which they're no longer connected. So shallower than that, these two basins are connected. So that's a really simple framework. Can it really work to explain those differences? Well, we started out by looking at a really high resolution model, a 12th of a degree model run for 50 years, which contains all sorts of physics and very energetic eddy fields and all the nasty nonlinear terms. And this is that boundary pressure. The red line shows that boundary pressure, the forces on the sides of the oceans as you go all the way around the global coastline. And there's a lot going on here, but what I really want to focus on is this difference between East Pacific, that's along here, and East Atlantic. There's 160 meters squared. That number is an equivalent sea level integrated over depth. So that's why we measure it in square meters. Incidentally, almost the same in the Indian Ocean as well as the Pacific. So that's our target. We want to explain that. And if we just use the winds part of our calculation, we get this blue line. And you see, it's worked pretty well, but we've only explained about 2/3 of that drop and doing a very poor job in the western side of the Atlantic. Well, we might expect that because we know there's a big overturning circulation in the Atlantic. And in fact, that accounts for this western part. But if we add that in, it makes no difference to anywhere else at all, just the West Atlantic. It's when we add the Indian and Pacific Ocean deep overturning that we find this. So I fix the Atlantic with 17 sphere drips of flow out below 1350 meters. But this match that we get now comes from the 2 and 4 sphere drops of flow into the Indian and Pacific Oceans. So it works very nicely, and we're getting to an accuracy of about 10 square meters, which translates to a couple of centimeters or less of sea level difference. So that's in a high resolution model, but does this work in the typical climate resolution models of the CMIP scenarios? Well, in short, the answer is yes. So here we're looking at predicted change in E versus actual change in E. E is the part of the boundary pressure that's due to density changes. So we can use our theory to predict how E will change based on the winds and that deep overturning in the Indian and Pacific. And it actually produces a very good predictor of what is observed in the models. This plot on the right shows that a lot of that change in E is due to the overturning. Not all of it. You can see the points don't all lie on this straight line, which they would if it was all due to the overturning. So the winds are playing some role, but actually it's by far the biggest contributor. In these models, what we see is that flow into the Indian and Pacific Oceans below 1350 meters is falling or even reversing by about up to 7 sphere drops, depending on which model you look at. Finally, let's leap as far as sea level. So we're measuring E, which is something to do with density, but the sea level is a different thing to do with density. So these things don't match up perfectly. But it does make a good predictor of a large part of that sea level variability. You can see the different models are doing slightly different things, but they're all aligning along this stretched out cloud. So a large part of that sea level change, which is actually to do with upper layer density, is resulting from these deep flows. So to summarize, although the sea level difference is controlled by this upper layer density, it looks like a surface process. This deep Indo-Pacific overturning is actually controlling what that density is. So these deep flows matter. The models predict a reduction in that sea level difference into the future, but there's wide variation in the models. And boundary pressures are a really sensitive way of monitoring what's going on in this deep ocean, even if we measured near the surface. So for the future, we must focus on understanding and measuring these deep circulations using this simplified framework and find out what the Atlantic overturning and Southern Ocean do within that framework, how they contribute. Now, I've framed all of this in terms of sea level. The overturning is linked to sea level variability, but it's also linked to many other things like the transport of heat, CO2, and nutrients. So understanding this overturning is very important for linking into the biological side of things and understanding the changes and shifting ecology, because you can see how these deep flows are affecting the shallow flows, which are transporting nutrients around it in all sorts of important ways. Okay, I'll finish there. Thank you.
Well, thank you, Chris. That's very interesting. And can I just check with Katie quickly before I start? Is my audio feed still good?
Yes, you're sounding good.
Okay. As we're talking about Liverpool being very cold right today, my heating kicked in and it's super noisy. But hopefully that's not going to make up too much noise on the recording. Okay, let me just share my screen. So I'm going to finish off today's live event with a discussion about how this all then relates back into carbon storage. So the very end of that that journey from cells to the climate, through productivity and then into the carbon storage that it corresponds to. Okay. So, hi everyone, you've met me at the beginning. I'm Jamie Wilson. I'm a lecturer in ocean sciences at the University of Liverpool. So I'm a modeler. I use models to look at the way in which biology interacts with carbon and impacts the carbon cycle in the ocean. So today I'm going to talk about the role that marine biology plays in storing carbon in the ocean and how we think that's going to change in the future. So the picture on the slide here is a kind of typical image you get from the deep ocean and showing the kind of weird and wonderful stuff we get in the deep ocean, such as this kind of funky looking vampire squid. But what I'm going to talk to you today is about what's going on in the background. And these are this tiny little white specks you can see all around this vampire squid in the background. So those white specks are what we call marine snow. So that's the sinking remains of dead plankton and dead organisms that's sinking down from the surface ocean where they were living originally and now sinking down into the deep ocean. So that marine snow contains all of the carbon that the phytoplankton fix during photosynthesis in the surface ocean. As it sinks, that material gets broken down by bacteria, consumed by other organisms, and that releases the carbon back into the ocean. So we have this net transport of carbon from the surface ocean, where it can interact with the atmosphere, down to the deep ocean, where it's now isolated away from the atmosphere. So once that carbon is in the deep ocean from marine snow, and it's been degraded by organisms, it will be mixed back up to the atmosphere and to the surface ocean. So that completes the cycle by ocean circulation. But that takes quite a long time, so anywhere between 10 to 1,000 years. And it depends on basically where that marine snow, how far that gets into deep ocean. So the further it gets and leaves its carbon, the longer it will take for it to get back out in the ocean. So we call this process the biological carbon pump. Biological because it's driven by biology, and a pump because it's pumping carbon from the surface to the deep ocean. And because that marine snow sinks on the order of kind of days to weeks, and the return flow of that carbon back to the atmosphere is on the order of 10 to thousands of years, we end up with this carbon that's accumulating in the deep ocean. So if we were to kill off all of the marine biology in the surface, what would happen is that carbon that they're storing would slowly leak out to the atmosphere, and that would increase atmospheric CO2, equivalent to the amount that we've increased it across the 20th century from human emissions. So why do we care about this? So this is a reservoir of carbon that's on our planet's surface, and it's potentially vulnerable to a change in climate, which would mean if it decreases in size, that would exacerbate our warming climate. So for context, I'm showing you here an assessment of all the different carbon pools in our Earth's surface, split by ocean and land. And on land, we're very concerned about losing forests and rainforests, for example, for the same reason that would increase the CO2 back into the atmosphere. And this is just showing you the magnitude of that impact if that reservoir collapses and the likelihood that we think that might happen. And you can see that the biological carbon in the ocean is a very similar kind of magnitude and uncertainty to that the carbon on land that we're worried about losing. So we're also interested in this because there's a lot of focus about whether we could actually enhance this process rather than decrease it. So by adding nutrients into the surface ocean, such as iron, we can stimulate more biological activity, stimulate more of that marine snow to sink down and store carbon into the ocean. So this is a process that we are interested in monitoring as a natural process, but also artificially enhancing to tackle climate change. So the emerging challenge in this area for scientists working on this is that this is actually really, really complex. So we have a huge range of ecological and biological processes that are affecting this marine snow. So we have various things swimming down, eating it, zooplankton pooing out very dense particles. So we have tons and tons of mechanisms affecting this marine snow. So to give you an example, on the left-hand side here, we've got some images of that marine snow from different parts of the ocean. So on the left-hand side here, you can see that those particles look very dense. They're very densely packed in. So that means that they're heavier and they sink much faster. So more of that carbon and more of that marine snow reaches down to the deep ocean and stores more carbon. In other parts of the ocean, you can see on this side, this material is much more gelatinous. This stuff is being worked through by ecosystems much more. So we end up with this little bits of cruddy stuff all glued together with this kind of mucus. So that's really buoyant. And that means that that ecosystem doesn't deliver that much carbon down to the deep ocean because it doesn't sink that fast. So the challenge for us is that biodiversity of plankton and their ecosystems is a key part of that vulnerability of this carbon pool. So the key challenge in understanding this is that our observations are really uncertain. So we can measure that marine snow using sediment traps. So this is basically a giant funnel that we stick out in the ocean somewhere, and it collects the marine snow into these bottles, and we can retrieve that and measure how much carbon is sunk to a particular depth. So when we've compiled all the observations we've made over the last few decades, we can make these maps that show where that marine snow and carbon is reaching, or how much is reaching the deep ocean. So this map in the yellow color shows where more carbon is reaching the deep ocean to be stored. And hopefully you can see that they effectively completely disagree with each other. So these are two different types of sediment traps at different parts of the ocean water column, both completely, as far as we know, completely correct. So the map on the left suggests more carbons reaching the deep ocean in the low latitudes, and that's consistent with that idea that dense particles are created in these regions. But the map on the left, on the right-hand side, would completely challenge this. So at the moment, we're kind of really uncertain as to what processes are really dominating this marine snow. So that has a big effect on our model predictions of how this will change in the future. So this is some results from a research project I did recently, looking at how the biological carbon pump changes in the model projections that underpin IPCC reports. So in this time series plot here, I'm showing you just the change on the marine snow reaching the deep ocean across the 19th and 20th centuries and into a kind of climate change scenario in the 21st century. And you can see that in the 21st century, as the climate warms, we have this big divergence in what models are predicting. So some are predicting a big decrease in this marine snow. So that would mean that this carbon pool is potentially decreasing. Some models are actually starting to increase, so perhaps strengthening this carbon pool. And this is a consequence of that uncertainty in our observations. So if we look at what those models predict in the same way as our observations, so the yellow color here is showing you where more marine carbon is reaching, but now in each of these models, you can see they're massively variable. And that's because these models are taking a best guess as to what process is driving that marine snow. And that best guess is not the same across different models. So hence why we have this really big divergence and uncertainty in predictions. So what's the effect on the actual carbon storage? So we looked at that as well in these model simulations. And we found that actually the opposite to what we might expect. So over that time period, we start to see the carbon storage in the ocean is actually increasing consistently across all models. So that's kind of opposite to what we expect because the biological fluxes are decreasing on average in these models. So why is that happening? So if we look at the ocean circulation is warming, it's slowing down. That rate at which the biological carbon is lost back to the atmosphere is slowing down. So we're holding on to that carbon that's in the deep ocean for a little bit longer. And that is overriding that loss of carbon in marine snow. And it's causing, in the 21st century, the carbon storage to increase consistently. And that kind of makes sense if we think about those ocean timescales. That's the longest timescales in this process. So that's really dictating what's going on here. So it's not been long enough in these simulations to look for or to see what the impact of those changes in biodiversity might be. So where does this leave us with biological carbon in the ocean and biodiversity? Is it really that important? So I would argue, yes, it is, but the question is we need to redefine the problem. So this is some recent work from some colleagues at GMR in Germany who looked at a model simulation of where we're reducing carbon emissions to the atmosphere. And so this is the time from the main reduction. And as we start reducing CO2 in the atmosphere, the anthropogenic CO2 that's stored in the ocean rapidly declines. And the biological carbon in blue kind of slowly just ekes up. It's just slowly responding, consistent with that idea that ocean circulation, which is happening over hundreds of years, is dictating the really long time scale of this. And at some point, few hundred years afterwards, this becomes a dominant source of carbon storage in the ocean, not anthropogenic carbon. So really, we need to reframe this problem in a different way. So we need to think of this as in a similar way to sea level rise. So we're committed to changes that are driven by climate change right now in this biological carbon pool. But we need to think about the impact emerging down the line in 100 years, 200 years, beyond the time that our generation, and that will then impact us in terms of our society and that carbon storage. So if we put the range of that carbon storage that we observed in the model simulations by 2100, you can see that range is massive. So that could mean this carbon pool is contributing loads to storing carbon in the ocean. It could be that it's contributing nothing. And we won't know that until hundreds of years down the line. So at Liverpool, I'm leading a new research lab funded by UKRI Future Leaders Fellowship to really tackle that question of the future biological pump. So we're leaning into that modeling aspect. So we're compiling the existing model simulations to make an accessible repository for the wider community to access and to do a comprehensive look at what's happening in this process in the future. And then we're running new state-of-the-art long-term simulations beyond 2100 to look at how this carbon storage might change beyond that. and whether the ocean physics and the circulation is always going to drive it, or whether we see those changes in the biodiversity actually kicking in and driving what's going to happen in the future of this carbon pool. So I'll finish there and just say thank you for listening. Okay. So that concludes all of the talks today in the session. If we have any questions, I wasn't looking whilst I was talking. We've got a couple of minutes, I think, for questions, and then we'll wrap up.
Jamie, can I ask a question?
You can.
The biological store of carbon, you're showing that's increasing. So that's not due to the source, but it's due to the sink, is it? And is that then linked to the amount of oxygen and the ventilation of the waters, or it's returned to the sea surface?
So there's two things there. So you're right in that we're seeing a decrease in how much carbon is reaching the deep ocean from marine snow, but the ocean circulation's slowing down at the same time. So it's holding on to that carbon that's already there for much longer, and that's overriding this kind of signal. So that also has an effect on the oxygen. So this degradation of organic material will use up oxygen in the ocean interior as it's sinking down. So the same is true for oxygen. So as the ocean circulation slows down, that should be exacerbating that low oxygen, the low oxygen minimum zones that we have in the ocean. The flip side, and what we don't know, is whether that oxygen will then affect the degradation of that marine snow, because we know that organisms need the oxygen to degrade it. So that might have an additional feedback that we don't know. So that's one of the kind of focus of looking at in our research project.
Thanks very much.
Okay. So I think we're at the end of the event. So Katie's urging me to wrap up. So I'd just like to say thank you for joining us today. Thank you to all the speakers who took part. Any questions? I'm sure the speakers will be happy to chat to you offline, via email, etc. And certainly myself, happy to do that. So again, just like to finish off and say thank you for listening today and thank you for joining us.